Good morning or good afternoon. Welcome to the first seminar of the Going Organic seminar series hosted by Clemson University. Today we'll have our first guest speaker, which will start with an introduction from Dr. Dil Thavaraja, and then we'll have the presentation, and then we'll open the floor to questions at the end. If you could please use the chat box or the Q&A box to ask any of your questions, and we will use the remaining time following the presentation to answer those questions. So now I will hand it over to Dr. Dil Thavaraja.
Thank you, Tristan. Good morning, good afternoon, good night. All over the world, people joining, our colleagues joining for Going Organic seminar series. My name is Dil Thavaraja. I am the PI of the project, Going Organic. We are breeding peas and lentil for biofortification, increased nutritional content, adaptation for South Carolina, but the work that we do would help for the older parts around the world. Co-PI, I like to acknowledge the co-PI of the project, Dr. Lucas Bortwright. He's the assistant professor at the PES, the same department. He's the bioinformatician. So we are hoping to develop a lot of genomic tools for this peas and lentils. And also the project manager is Tristan Lawrence, and you will hear from him a lot on the updates of the Going Organic website and the institute that we are conducting in the summer for the breeders and the students. Before we start, I like to acknowledge the partners of the project, the Good Food Institute. I acknowledge the Carolina Farm Stewards, especially Mr. Mark Dembski, E-Extension Platform, WP Rolls and Sons, our own farmers, locations for field trials, Living Web Farm in North Carolina, and I would gracefully acknowledge USDA NIFA OREI for funding the phase 2 of the lentil and pea breeding. Today, it's my great honor to introduce Dr. Asha Karunaratne, one of my former colleagues when I was an undergraduate student. Dr. Karunaratne is a professor in crop production system modeling at the Faculty of Agricultural Science, Sabaragamuwa University of Sri Lanka. She obtained her BSc Honours from University of Peradeniya, Sri Lanka. And also she has an MPhil from University of Peradeniya. She obtained her Ph.D. from University of Nottingham, UK. And she has already worked in the Walker Institute for Climate System Research, University of Reading, and the University of Nottingham, UK. Currently, Dr. Karunaratne is the Fulbright Fellow at the University of Florida, Gainesville. She has worked more than 23 years on the quantitative crop physiology, focusing on crop yield responses to variable climate and crop simulation models. We are expecting to collaborate on this crop simulating models and how it fit into the organic models. So that's why we invited Dr. Karunaratne to share her experience, and especially she's going to talk about the Bambara groundnut, how it adapted into the system. So thank you so much, Dr. Asha, joining with this. And now the floor is yours. Welcome.
Good morning, good evening, and good night to all of you. And thank you very much, Dr. Dil, introducing me to your research community, Going Organic. And indeed, it's a pleasure for me to do this talk today as a guest speaker to the Going Organic community in your research group. Today, I'm going to basically talk about the potential of underutilized crops for resilience of farming systems to adapt climate change. The presentation is aligned with two case studies. One is a leguminous crop, Bambara groundnut, which is Vigna subterranea. And the other case is I will be presenting with cereal, which is proso millet, Panicum miliaceum. I think I don't have to tell about myself. Dr. Dil already explained. So my current, this time of the year, I am at the University of Florida as a visiting scholar. I'm working with the Institute for Sustainable Food Systems with Dr. Hugenbaum, developing a crop model for Bambara groundnut. I will talk about my current work also during this presentation. And this is my university in Sri Lanka, Sabaragamuwa University, which is situated in picturesque mountain views of Belihulloya, southeast part of the country. There you can see my university. And I'm a professor at the Faculty of Agricultural Sciences. This is my Google profile if you want to explore more. And at this stage, I want to introduce my collaborators to the current work. The University of Nottingham School of Biosciences, Neil Crowd, Professor in Environmental Modeling, with whom I did my PhD. And Sean Mays is the molecular genetics or the molecular breeder of Bambara groundnut, who was a part of that project. And this is Sean Mays, where we did some field observations of the glass house experiments at Nottingham. And my other main collaborator is the University of Crops for the Future Research Center, which is hosted at the University of Nottingham, Malaysia campus in Kuala Lumpur. Actually, this year it has moved to UK as the same center. This is the Global Alliance for Underutilized Crops. Professor Saeed Assam Ali, who is the CEO of this center, is one of my leading collaborators, this Bambara groundnut research, and he is like a pioneer of underutilized crops research. And I have some collaborations with a professor from the University of Bloemfontein, South Africa, Sue Walker. This is Sue Walker. She is a professor in agrometeorology. And the other collaborators are from the Agriculture and Model Intercomparison Project. Alex Rouhani is the PI of climate team. So with all these collaborators, I have done some work for modeling the Bambara groundnut and proso millet to evaluate its sensitivity to the climate through models. Okay, the problem. The whole world is depending upon this tip of this triangle with too few species. This has imposed a reasonable risk, volatility, and also the overdependence of the selected few species. So the intensified agricultural systems have reported the constant reliance on 30 major crop species to fulfill the global food demand. However, around the world, about 7,000 species have been extensively harvested and utilized for food, but they are not considered extensively in research, as well as the information are very localized, and we call them as underutilized species. However, this overdependence and the narrowing of our food basket has led to some lifestyle diseases. And the main question that comes is, are we fit for the future climate with only these too few species? So therefore, the alternatives, so-called underutilized crops, have the potential to meet the challenges in the developing world, especially providing agricultural diversification as well as dietary diversification, also fit into the climate through showing more resilience to changing climates that will come up in future. So the solution is broaden the world's agricultural biodiversity. As you see, this green box here, we want to broaden the agricultural biodiversity through innovative approaches. So the approach that we follow are identify, improve, and use more species. And we use the experience gained on major crops like soybean, rice, maize, wheat, etc. And this is tested and extrapolate using the crop models. So then we can check whether it's fit for the future climates. Okay, now we will begin the story about Bambara groundnut. This is an African protein-rich legume grown by subsistence-level women farmers in sub-Saharan Africa. The cultivated crop exists as a landrace and grown extensively at the subsistence level farming systems. I hope now all of you are familiar what is a landrace. It's not an improved cultivar. A landrace is a locally adapted strain of a species that has been selected through traditional methods and not influenced by modern breeding technologies. So, and this crop has been grown throughout Africa, from Senegal across to Kenya, from the Sahara to South Africa, and was expanded to Indonesia and India to some extent. And traditionally, this is grown by the women as an intercrop, and where other crops fail, because this can withstand severe drought as well as the heat stress. Compared to other legumes like groundnut, it survives in a drought conditions. The origin is mainly the somewhere around Cameroon region. So this the lady here selling different types of legumes. The one tin of Bambara groundnut is much more higher than the other legumes. So that's what we observed when we had some field visits to those countries where Bambara groundnut is specifically utilizing. This is how the crop looks like when it is uprooted. The pods come just below the soil. In 2001, the first global mapping of yield was done through the GIS experts in FAO. And this map explains the yield prediction. That was like a very basic modeling exercise. It was not a very dynamic model that used for this upscaling. However, this gave an indication that it can be grown in some other parts beyond the African region. So that has been taken into account, and through EU projects, it has extended to Indonesia, in West Java and East Java. Now it's really growing to higher extent. This field is from one of the Indonesian partners. Okay, with this introduction to the crop, I want to give an overview of a crop model and its applications. The crop model is based on the understanding of plants, soil, weather, as well as the management interactions. So in terms of morphological, phenological development, the crop is the information in terms of morphological, phenological, and development aspects are considered. And also the photosynthesis, respiration, growth, root water uptake, stress, all the physiological aspects are considered in the crop models. And it is converted into some mathematical functions and predict growth, yield, and timing. The main input to the models are field characteristics like climate, soil, and the cultivar characteristics, we call them as genetic coefficients, and the management, the planting dates, the fertilizer application, rates, timing, irrigation, organic matter management, crop residue management, etc. They all are utilized in the model. And also this can be used to perform what-if experiments. This is where we can think of linking to this organic group in Clemson University, because you all are breeders. So you can have some pre-decisions using models for those lentils and peas with what if experiments run through the models. You don't have to invest a lot of field work to that. The The only issue is we need to develop models for those crops. So currently I am at the University of Florida with the team who developed this decision support system for agrotechnology transfer, which is called DSAT. This is a world-renowned crop model that has been utilized by modelers around the world. There are more than 4,000 utilizers, I guess. So what I'm doing at the moment is I'm developing a new model for Bambara groundnut to integrate with this DSAT interface. They have done series of legumes like peanut, faba bean, soybean, chickpea, etc. in terms of legumes. But there are many other crops, cereals, tuber crops, different expertise have contributed. So my expertise I'm contributing to develop the crop model for Bambara groundnut for DSAT. Actually, the work has been completed over the last 3 months. Now we are in the writing up stage. So before that, I did a simple crop model for Bambara groundnut as my first step in modeling. That is called BAMGROW. I have published it, actually it's my PhD work. I have published that in the Experimental Agriculture Journal. That also has some routines integrated, like we have the weather, temperature, crop growth, photoperiod, and soil water. So these are the input parameters, as I mentioned earlier. It's more or less common across most of the crop models. And this model, through this model, I could predict the yield, the canopy, and also the partitioning of dry matter to different components. This was based on the most well-established model for legumes, that is CropGrow, which is used initially for peanut. I used most of the routine of CropGrow peanut to develop my BAMGROW model, and that has become a success story in my modeling career. More about the crop phenology. The Bambara groundnut shows partial indeterminacy. That is the problem that we are facing at the moment to indicate its maturity and harvesting time. You see, the crop life cycle span from 90 to 150 days. This is the variation among the land races. So there are very short duration land races as well as long duration ones. So this is again a problem. The diversity, very high variability among land races is a challenge for the crop modelers, unlike in very high level improved cultivars, because cultivars having the very defined phenology. Usually Bambara groundnut germinates between 7 to 10 days, and flowering starts between 30 to 45 days, and the pod set 15 to 40 days. And there's an overlapping of flowering and pod setting, and the pod filling and flowering continues until harvesting. That is another challenge. So at the moment, we don't have any information that we have not recorded any of our experiment, the first seed appearance, the first flowering to first seed, the time duration, and also the harvest maturity. It is very difficult to detect due to this partial indeterminacy. This is where I conducted most of my experiments at the Tropical Crops Research Unit, University of Nottingham. It's the Sutton Bonington campus, we call as the School of Biosciences is there. There are 5 glass houses that I worked all of my 3 summer months from 2006 to 2009. I was inside these glass houses during my PhD. And this is the setup inside the glass house. We have everything controlled, the temperature, the monitoring of solar radiation, and there are some drip irrigation systems to provide the irrigation. So we had some irrigation trials and temperature stress trials and many more within this glasshouse complex. So these are the series of experimental data that are used for developing the model and also calibrating. In 2000, they have done, this is not mine, it's by some of my former colleagues at Sutton Bonington, but they collaborated with me providing the data. So there's a growth room experiment that was conducted for evaluating 10 land races for 5 temperature regimes. And in 2002 and 3, 2 land races were tested for irrigated and drought conditions under 28 degrees. This is considered as the optimum range of temperature. And 2006, which was my experiment, we imposed 2 temperature levels, low and high, 23 and 33, for the same 2 land races from previous experiments. And 2007, in 2006, actually, only the temperature we provided it was fully irrigated. In 2007, the same 2 temperature levels, but we imposed late drought to see the interactions of temperature and drought. And 2008 was the worst. It was early drought plus temperature. So these data are very detailed measurement data available that are used for models. So through this growth room experiment, this is actually the observed rate of leaf appearance from these 10 land races. And except this 35 degrees, when increasing the temperature, it has an increase in trend of rate of leaf appearance. At 35 degrees, all the land races explained a reduction of rate of leaf appearance. So in DSAT, I set up an experiment through environmental modifications. There's a module that you can, there's a tool that you can use environmental modifications. That is the new technique that we can use to evaluate these. We can evaluate the climate change impact using that environmental modification tool. So I impose all these 5 temperatures. This blue line is my model prediction, and it was compared with the mean value of this observed data. You see the variation, it's very high, but the model is quite satisfactory. And not only this temperature variation or the response, and I want to highlight the drought tolerance capacities of Bambara groundnut, which is another significant feature. So based on the literature, it invests heavily in its root system, not even drought, but in generally compared to the peanut. And it has different pressure volume water relations compared to other legumes. And it comes to more negative with the drought accumulating more solutes, and also the leaf water potential, relative water content remains stable until a critical point. So it can maintain that relative water content stable. At a certain point, it collapses suddenly. So the crop dies after it comes to a very critical stage only. Until then, it can withstand. And there are some waxy layers in the leaf, so it exhibits the lock and key mechanism for the canopy conductance. And also there are low stomatal density. Sometimes it's a resilience mechanism to resist the drought. So these are some of the current work that I did with this set. I use 2 land races. This is just an illustration of a particular one land race. This is the canopy. In terms of canopy, the leaf area index is considered as an indicator. So you see these 3 colored lines, green, red, and blue. The green one is the high temperature. red is the optimum temperature, blue is the lower temperature, and the model prediction are the solid lines. So you can see it goes along with the actually observed measurements. So the statistics of the model prediction is at a very reasonable level. The root mean square error is less than 0.5 in all the 3 treatments, and with the D-stat value is more than 90, which is highly acceptable. And the pod weight is vice versa. So the pod weight at optimum temperature is having the higher, while it is lower with high temperature, but it's not that different to the low temperature. So again, here the model prediction is reasonable, and the root mean square error is less than 500 kilograms per hectare, and the D-stat is more than even 95%. So this was previously extended. I have done some more extensive work with evaluating the Bambara groundnut for climate resilience. The study sites that used for this study was Gaborone, Botswana, Bloemfontein, South Africa, and Caprivi in Namibia. As you see in this map, these colored areas are the 3 study sites. Orange is Namibia. This is Gaborone, Botswana. Bloemfontein is in South Africa. So we tested 2 land races for these 3 locations in Southern Africa. But in terms of climate, the Botswanian climate is similar to Caprivi, but slightly drier. In South Africa, Bloemfontein has a cold, semi-arid climate with sunny, dry days, even during the winter growing season due to high altitude. This contrasts with the hot, semi-arid climate in both Gaborone and Caprivi. So we could capture the suitability or the adaptation of land races to these different diverse climatic regions in Southern Africa. The climate projections were done with this AgLib climate team, especially Alex Ruani, who helped us to downscale the climate scenarios for RCP 8.5 with 20 GCMs. And we evaluated the performance of those 2 land races for mid-century, 2040 to 2069. Before that, we always have to consider the baseline climate. Baseline climate was considered from the observed data from these 3 locations I mentioned earlier, and their data were taken as the baseline climatic conditions. So in addition to that, I did additional work with the Coordinated Climate Crop Modeling Project, which is C3MP, which is again a part of AgNIP. This is very common now. Maybe you can search it in the AgNIP website. So in here, the climate sensitivities were tested with these metrics. So there are 3 parameters: temperature, precipitation, and carbon dioxide. The lower bound is considered for temperature -1, and it goes up to +8. The precipitation -50 up to 50%, +50. And the carbon dioxide concentration from 330 to 900. So these metrics has been used to produce 99 climate sensitivities. So this was tested, and we got the results for these 2 land races. So this is the climate projection basically for those 3 locations. The blue star is the baseline climate in Botswana. When it comes to the mid-century, we're using this using RCP 8.5 scenario and 20 GCMs, it has increased a lot. In South Africa, it's much more. In Namibia, it's not that great, but it has already increased. The seasonal rainfall change due to the climate change was evaluated using this predicted downscale climate. Here, the red star is the baseline precipitation. It's not that differ, except in Namibia, it shows some reduction in the rainfall pattern. These are the yield predictions for those 3 locations. This is basically the baseline. The left figure is for the baseline period. 2 colors are for 2 land races. In Namibia, the orange land race shows a very high yield because that is a Namibian land race which has been originated from that location. So it has some site-specific adaptations. And when it comes to the future climate, we evaluated the changes of the yield as a percentage due to the GCMs. This dotted line is the reference for the zero change. And you can see all 3 sites have yield gains for both land races. And in South Africa, this orange Namibian land race S19 has gained a very high advantage due to the warming because Bloemfontein is considered as a cold climatic region. So it has an advantage of this. So this is the results. These are the results of these 99 climate sensitivities. These diagrams are emulated diagrams considering now, for example, if you concentrate to the first box of the top left hand, here the x-axis is changing in temperature, y-axis is the changes in precipitation, where we keep the carbon dioxide concentration constant. So likewise, this is considered as a 3-dimensional sensitivity. The star is for the baseline. The more green is more better yield, brown is less yield. So that is how it explains this sensitivity test. So based on these results, I can say that Bambara Groundnut reported diverse yield performance with diverse sensitivities to climate between land races originating from 2 locations. These 2 sets are for 2 land races. S19, which is originated from hot, dry conditions in Namibia, has a greater yield potential in future compared to the Swaziland, which is Swaziland is considered as a cool, wet climate. South Africa has the lowest yield with current climate, indicating positive yield trends in future. And Namibia reported the highest baseline yield with optimum current temperatures, indicating less potentials in future. So based on that, we can say that Bambara Groundnut holds yield potential up to 31 degrees, and further warming is pushing yields down. So there are many regions in Southern Africa that therefore can be utilized this crop successfully in coming decades. So this modeling exercise specifically provides some decisions for matching the genotypic suitability for a climate, for a region, considering a specific location. So this is our paper that we published out of that work. So it's in the Climate Research Journal. Okay, that is the story about Bambara Groundnut. Now I move on to my work in Sri Lanka. Actually, this was again a part of a PhD program from the University of Nottingham, Malaysia campus. A student completed the PhD about 2 years ago. And now he is a staff member in my department, and he is contributing some of the modeling work in my research team. He is Dr. Vimalasiri. So the work that I'm talking is prosomillet, which is, I think I already introduced, is Panicum miliaceum. This is our map of Sri Lanka. As you see, this ash color box is the region that we identified for this study as the proso-millet growing regions, but it has been, extended beyond this limit. So this is also a underutilized minor millet. It is cultivated some parts of the country as well as it's a crop in India, and I think it's the major molecular type research are being conducted at the University of Cambridge in UK, and also Sean Mays from Nottingham University is contributing to some of the molecular work with Cambridge, and we provided some genetic DNA extracts from our accessions from Sri Lanka to do the sequencing from that side. So we have some links with Nottingham University in terms of this work. This is the principal crop during the dry season. The yellow season in Sri Lanka is basically the dry season, and it's grown in, we call this the dry zone in Sri Lanka. It's a good substitute for rice, and the climate sensitivity, and the future yield. Crops like proso-millet receive less research attention. That's why we wanted to do this work. So this is the picture of proso-millet. It's more or less like rice. The panicle is exactly like rice, and it's highly nutritious. You can use it for making several types of food, and it's a bird food as well. So this is considered as a crops for the future because it requires low water. You can grow in between two seasons, and also the areas where rainfall is very little, and it can tolerate heat, and also no need very improved soil conditions. However, it's highly nutritious. The protein content is very high, and the economic value is high, but farmers are not that interested to grow in this. So that is another area to investigate, because there are some issues with the seed source as we identified. So they always go for the common crops rather than growing this type of nutritious food. So during Dr. Eranga's PhD study, he evaluated the onset and the retreat of the growing season of those proso-millet growing regions. For that, we got some met station data from these locations, and it was analyzed, and he came up with the date of sowing, panicle initiation, flowering, and harvesting. It was quite deviated from what farmers usually practice in. So that is another issue. The farmers consider the onset based on their traditions, but the real onset is different to the practiced onset. There again, we did this climate sensitivity test with the 99 sensitivities following the C3MP ACNIC protocol. They are actually, we use this all 5 station weather data, and also 5 accessions were used for the farmer selected varieties. And it shows the yield did not change up to 2 degrees change increment of temperature. So that is also an advantage, because now with the climate change, it is predicted 3-degree rise in temperature in most of the places, but this has some advantage up to 2 degrees Celsius centigrade. The yield predictions for this study, we evaluated 2 emission scenarios, RCP 4.5 and RCP 8.5. And according to the results, the simulated yield for mid-century were not significantly different or deviated from the baseline, so there's no reduction. An average of 5 proso-millet accessions increased 20% from all the GCMs under RCP 4.5 scenario. So that has some advantage. So the current ongoing work, we have an MPhil student. She's also a staff member of my department. But we are trying to look at this morphological and molecular characterization of prosumilate landraces cultivated in Sri Lanka. These are the objectives of that MPhil study: to analyze the genetic diversity, to characterize the agronomic traits, so then identify superior genotypes for improvements. So we link this work with Nottingham, Sean Mays. So he has given us some guidance for developing SSR markers and provide some supervision to this MPhil student. The problem statement is up to date. There is no proper research evidence related to crop diversity of prosumilate landraces, and they have not been contributed to any breeding programs due to the poor availability of morphological and molecular data. So that is why we wanted to pick this. So these are the current locations that we target to collect samples. Actually, we have already collected. Due to the COVID-19 pandemic, we had to stop some of the field work. So the mature seed samples are collected from farmer fields from different agroclimatic regions in the country at the end of each growing season. When the farmers are about to harvest, we go there and collect some samples. This is one of such samples that has been collected from this northern part of the country. So then we try to multiply them to produce 4 to 5 generations, and the experiment will be laid out in complete block designs. I think now the experiment should be going on in the faculty farm. So after that, the next step is molecular characterization, the DNA isolation from leaves, and then PCR amplification following the SSR primer sets. So this will follow to the phenotypic characterization using some qualitative traits and quantitative traits. So that's what I want to talk about. This is again my university. We celebrated our 25th anniversary this year. So you can see the picturesque mountain view of Ballyhulloe, where my university is situated. Thank you very much.
Thank you so much, Dr. Asha Karunaratne, sharing your research, and it's very informative. So there's questions coming into the Q&A. Please put into the chat box or the question, and then I will start discussing because we have a couple of more minutes left for the questions. So there's the first question coming from our Rwanda partner, Dr. Hilda. And she is wondering about how about the amino acid pattern in Bambara nut? Have you studied that among the 2 types of landraces? She's wondering about the amino acid profiles.
Yeah, that's an interesting area, but actually that is beyond our research focus. So we have not done. Even I wanted to get some idea about the nitrogen levels in leaves for calibrating the DSAT model. I digged into some of the literature to find, but it's not specifically to those 2 land races. They have been done some other land races. No, that was not done in our research.
So a follow-up question for that, what is the protein content of this Bambara ground?
It varies again with the land race. It comes around 10 to 15%.
Okay, so do you know what's the fat content?
It's not that high as I got from the published information. It's quite lower than the peanut. It's about 3%.
Okay, so in your experience, you think Bambara ground is more climate resilient because of their moderate fat and protein balance?
I'm not really expert to comment on that because we have not done any proximate analysis or biochemical research in terms of that. But other than that, what we understood as physiologists, so they have some physiological mechanisms to resist the drought and heat. So that's how it shows some adaptations.
Oh, that's interesting. There is another question from our Indian partner, Dr. Samuel, and he is wondering about organic study in Bambara ground. So he's asking, what's your impression about adapting Bambara groundnut into the organic system?
That is very possible because this requires very little input and you don't have to add any fertilizer, especially. And also you don't need, there is actually no any pest disease incidences. So you don't have to worry about spraying anything. So it's very suitable for organic systems, as well as it's a good crop for intercropping. And in between two seasons, this can be used as a between two season crop as well.
Oh, that's very interesting. Also, he is wondering, Dr. Samuel is also a plant breeder. He is also wondering how much this is practiced in Africa. I see in the participation, some of our African partners are in there as well. So he's wondering how much this is grown in the other parts of Africa, other than the countries you mentioned.
Oh, it's grown in most of the drier parts of Africa, not only these three countries, Botswana, Namibia, South Africa, like they do at research level. It's in Tanzania and Malawi, Nigeria. I have come across series of papers. Some I'm getting for reviews. Everything comes when Bambara groundnut comes to me for review because I have been published a lot. So it's grown in many parts of Africa. It's a crop of African, interest. And now it has spread to India as well, to a lot.
Right. I've seen in Ghana as well as one of the leading crop.
Yeah, Ghana.
The cowpea and other things. And actually it tastes quite well. I like Bambara groundnut. So another question coming from Dr. Chumani from Clemson University, and he is actually a pathologist and wondering about whether you did any pathology studies, especially for nematodes.
We don't actually experience that big pathology issues regarding Bambara groundnuts. So it was not interested in any of the research partners. Especially more interest is towards this molecular genetics other than this pathology, because it's not a big problem actually.
Okay. All right. Yeah. It's very adaptable crops. So there's a lot to study as well. And Dr. Samuel, want to follow up question. He's wondering whether do you know anybody grow organically Bambara nut?
I think most of them are organic in Africa because the farmers are very poor farmers. They are subsistence. They don't put anything onto their soil except this. The rain comes from the sky. So other than that specifically, I can look for it, but I can't name right now any research group or anybody who is doing this Bambara groundnut as an organic team. Even the Nottingham team, we don't do any genetic manipulations. It's the developing SSR markers and doing some, try to release some varieties because at the moment there are no varieties. So something like that is going on. They are all organic, I guess, but it's not really labeled as organic.
Yeah. I totally understand. The organic system is low input. Like here, it's specified by USDA that we cannot apply unless it is USDA certified organic. But when I talk to the breeders around the world, they always telling me like, well, we are organic because we don't add much of the system. So like you're correct. And I think that's the beauty about having legumes because we can, it's a very minimum input into the system. So. And actually, this is the message from Dr. Hilda from Rwanda, and they are actually trying Bambara nut right now for their adaptability in Rwanda. So I'm actually hoping to visit her next year if the COVID goes away. So maybe we could make the connection and work continue working on Bambara ground in Rwanda. So that's very interesting. And there's another question from Mr. Tony Reader, one of the graduate students at the Clemson University, actually from my program. So he's wondering, you mentioned no instance of plant diseases. Was that solely from the plant grown in the greenhouse, or does that include observation from field-grown plants as well? So he's wondering whether, because you did not see these diseases, because it's grown in the greenhouse, he's wondering what it looked like in the field.
The greenhouse, actually my experience is mainly greenhouse, and there's no any incidence of pest or diseases, not only for Bambara groundnut, even for any crop, that is obvious. But I have been working with several African partners. My PhD was in EU Framework 7 project. So we had partners from Botswana, Ghana, Namibia, Tanzania, etc. As far as I can remember, no one was very interested in studying about these diseases or pests. So it shouldn't be a big problem even in the field, I guess.
Yeah, that's what I was thinking. That's what I was going to talk to Dr. Hilda as well. So maybe someday we can have a panel discussion about the African legumes. There's another breeders that are in our project. So, and there's another question coming from Dr. Shruti Narayana. She's the plant physiologist at the Clemson University, our department, and she's asking about, can you talk about the physiological mechanisms behind the drought and heat tolerance to Bambara groundnut? So, and then also you mentioned that you observed some interesting physiological mechanisms. She's wondering about any physiological mechanisms related to the Bambara ground.
Yeah, these are some of the mechanisms. Actually, what I summarized here is about as the results of about 20 years of research from the University of Nottingham conducted, imposing different drought levels, different irrigation conditions, and even not only that, photoperiod levels, etc. So it has a very high level of tolerance. This is because the cells can accumulate more solutes, so it can withstand the pressure or water potential without collapsing, without come to the plasmolysis of the cells. So then to reduction of the water potential, it has some tolerance. So that's what in terms of physiology. And the leaves having lock and key mechanism. So it has the canopy conductance. It controls the canopy conductance. In this slide, it explains all these drought tolerance mechanisms of the crop. And they have sunken tomatoes, which is again a conservation of water, mechanism of water conservation. And the partition into the roots is very high, not under drought. It's a general character. Actually, I consider that in developing the model for DSAT, because other crops like others have very less amount of dry matter partition into the roots. But when I wanted to do my crop, I explained to my host here, though the developers, so no, this crop behaves differently. We have to invest more on the roots, thereby it can withstand the drought stress. So these are some of the mechanisms, and there are series of papers have been published in drought mechanisms of Bambara groundnut.
That's very interesting. Thank you very much for that question. And this is my colleague from North Carolina, USDA, Dr. Chris Smith. And they are actually growing handful of cultivars for the second year in Western North Carolina. And he's curious if certain cultivars are better in cooler and shorter seasons. You know, our growing season, because I saw that you put in 90 to 150 days. So that's a long season.
Yeah, long variation. Yeah.
Yeah. So, but do you have a shorter season? Like most of the summers in, this is good for Saskatchewan weathers even, I know. And I am trying this for South Carolina as well. Our season is less than 100 days for winter. We can use this as a winter crop. So what do you think as a cooler, shorter season?
Yes, there are actually the two land races that I extensively studied. Out of those two also, the two explains two different growth durations. The Namibian one is quite shorter compared to the Swaziland one because it adapted to dry and hot. It completes the life cycle very quickly to avoid any harsh conditions. So it's shorter, the flowering quicker. So there are, the landrace variability is very high. So you can't pick. It's very possible. Shorter, like 90 days is quite short.
Yeah, maybe you will develop the markers and then that will be easy for the breeders. One last question. Actually, I was wondering, our population is increasing and then the COVID hit and mostly food security is an issue. So what do you think of the other legumes by using your models, how we can push this food security more like for your model? Would it possible for you to include some nutritional aspects like proteins, amino acids, like what mostly we do in our
Yeah, the current model that I'm working this at is having all these provisions for all these. You can include the nutritional aspects, the carbon recycling, nodulation, the nodule behavior, the contents, the nitrogen contents of nodules even we can consider. So that depends all your data. If you got data from all those things, it's very possible to incorporate within the model. And at this stage, I want to tell what we can collaborate in future is now I have worked with this crop and produced a new model for Bambara groundnut. So it's very possible for one of your crops, we can produce a model if you have the detailed data sets. That's the requirement, especially this genetic coefficient, to derive the genetic coefficient, soil characteristics, climate, management, etc. If the data is there, it's not a big deal. So it's very possible.
Thank you so much. So I would take this opportunity to thank you, Dr. Asha Karunaratne, coming all the way from Sri Lanka and located in Gainesville and giving us a wonderful presentation, sharing all your information and hoping most of our team will collaborate with you. And I also like to thank the NIFA, OREI, USDA for funding these projects and bringing these experts around the world. And please tune to our website. We align 13 to 15 guest speakers for next whole year. And you will listen from climate change to nutrition quality, to plant breeding, pest and diseases, everything. So this is a great opportunity for all of us to join, and thank you so much. And we hope to see you again in end of August.
Okay.
Thank you.
Thank you very much, Dr. Dale, for providing me the opportunity. Okay, bye.